Today I'm going to talk about my research, PhD research, about production of fumonisin by Aspergillus isolates associated with Australian wine grapes, implications for fumonisin in wine. Little bit of introduction. Fumonisin B1 and B2 considered as carcinogenic toxins. They belong to group 2B. As they are carcinogenic compounds, potentially, the fumonisin levels in human food, mainly corn-based food, is currently regulated by some authorities. According to European Union regulations, the maximum amount of fumonisin that can be contained in food is range between 0.2 to 4 milligram per kilogram of food. So this is the chemical structure for fumonisin B2, the most abundant fumonisin type produced by Aspergillus species is associated with grapes and grape-related products. So this slide is to show you the current research findings, related to occurrence of fumonisin on grapes and grape-related products. You can see that the amount of fumonisin present in grapes is lower than the amount present in dried grapes. And also you can see here that the amount present in wine is really low, very small. So here you can see the gap between the amount present in wine and grapes. So this indicates us that something is going on here, like during winemaking process, it may be degrade. However, we don't have research information, published data related to this. And also I want to mention here that most of this research has been conducted in Europe and countries around Europe. None of the research has been conducted in Australia or many other viticulture locations. However, the research related to ochratoxin production has been revealed that the production of ochratoxin is regulated by ecological factors such as temperature, water activity, that sort of factors. Therefore, we can assume that the fumonisin production can be vary based on the agroecological zones. Therefore, it is very important to identify the fumonisin production ability of Aspergillus species associated with other viticulture locations as well. Around this, I set up my objectives for the research. So these are my objectives. The first one is to characterize Aspergillus isolate collected from Australian wine grapes. The second one is to investigate fate of fumonisin during winemaking process. The third one is to study the effects of wine storage temperature on the stability of fumonisin in wine. So in the next slides, I'm going to show you like how I achieve these objectives, my experiments and my results. So the first here, the characterization of Aspergillus isolates. To achieve these objectives, I collected 44 isolates from vineyards in New South Wales, Australia. So this is the location where I collected my isolates. So I extracted toxins, fumonisin toxin from these isolates and analyzed this toxin using LC-MS. So I found that, according to my results, I found 6 isolates positive for fumonisin. So these isolates were identified using PCR species-specific primers. So they belong to Aspergillus niger and Aspergillus welwitschii. It's called as Aspergillus omari as well. So these are the fumonisin production ability of those isolates that I detected from Australian wine grapes. The first 3 isolates are Aspergillus niger isolates. So the other 3 are Aspergillus welwitschii or Aspergillus omari. So you can see that the fumonisin production is really higher in CYA media with 5% sodium chloride compared to synthetic grape juice medium. So, and also you can see the difference of the fumonisin production of the 3 isolates of Aspergillus niger and Aspergillus welwitschii. Aspergillus welwitschii isolate produce higher fumonisin in both media. However, previously, previous research has found that Aspergillus niger produce more fumonisin compared to Aspergillus welwitschii. But according to our finding, we found that Aspergillus welvichae isolates produce more fumonisin. So the next objective is the fate of fumonisin during vinification. To study this, I actually inoculated Chardonnay grapes collected from a vineyard in New South Wales with one of the Aspergillus welvichae spores of one of the Aspergillus welvichae isolates that was produced, found to be produce higher amount of fumonisin in synthetic grape juice medium. I inoculated them in grapes and incubated for 7 days. You can see in the picture, like after 3 days, the fungus has started grown, and after 7 days, it's sporulated very well. You can see the tiny dots, black dots, they are the spores of the Aspergillus. So what I did was I made 4 categories of infection, mixing healthy grapes with infected grapes, and made wine using these 4 categories. So these are my results. So what happened during what happened to fumonisin during vinification process? So the y-axis, you can see the mean fumonisin content, and the x-axis, you can see the percentage of infected berries, different levels. The first one is control, healthy grapes, and the 25%, 50%, and 100% infection levels. You can clearly see that the fumonisin amount has reduced from juice to wine. There's around 80% reduction of fumonisin from juice to wine. So this clearly shows that during winemaking process, some amount of fumonisin is removed. However, we don't know like it is actually removed or whether it is hidden, and we can't identify. At this stage, we can't answer this question. But at this stage, we know that clearly it has shown like reduction has happened. So in the next slide, I will show you what has happened to this fumonisin. So you can see that the percentage of infected berries, the first column, and the percentage of fumonisin partition in each stages. So you can see that in the solids, marked and lees has taken up most of the fumonisin, and only a very small portion has been passed on to juice and wine. So this gives an answer that actually the fumonisin is removed with solids like mark and lees. Most of fumonisin is removed, and only a small portion of fumonisin is passed on to juice and wine. It is very good for the wine industry, I guess, because we don't want to have fumonisin in our wine. So this is a really indication that very small portion of fumonisin is passed on to wine, because wine, this is similar to the ochratoxin. Ochratoxin also found to be removed during winemaking process. Most of it is taken up with solid like mark and lees. So in the next experiment, I studied stability of fumonisin B2 during wine storage, because some amount of fumonisin is passed on to wine. So we don't know like whether how long that stays in wine. So this experiment was set up to study how long they are stable in wine storage conditions. I studied in 2 different temperature, cellar conditions, around 17 degrees Celsius, and around 25 degrees, like the handling conditions like conducted in room temperatures. So I studied in these 2 temperatures. You can see that like it is almost stable in both temperatures, but 25 degrees, there's some reduction compared to 17 degrees, but during our experimental time, it was almost stable. So the experiment is continue like another 6 months. So I don't know whether it is going to be degraded after some time. So at this stage, it shows that fumonisin is a stable compound in wine. But based on previous findings, like fumonisin could be degraded in wine because of the acidity, the pH, but we didn't find any evidence. And also I analyzed this wine sample for any hydrolyzed compound. I couldn't detect any hydrolyzed compound. So at this stage, we can only see that they are almost stable compounds in these wine storage conditions. So these are the conclusions I made based on my results. Aspergillus niger and Aspergillus welvichae species are responsible for fumonisin production in Australian wine grapes. Aspergillus welvichae produce more fumonisin than Aspergillus niger. Fumonisin production in CYA media with 5% sodium chloride was higher than synthetic grape juice medium. This gives us an indication that they produce higher amount of fumonisin in low water activity media than grapes. Approximately 97% of fumonisin removed during solid separation process with mark and lees, and around 0.5% fumonisin was passed to wine. And also we found that fumonisin B2 was stable in white wine at least for 5 months at wine storage conditions. Finally, I want to acknowledge these people who helped me with this research. Thank you.
Okay, thanks for your nice presentation. Is there any question? Joseph?
I have one question. Yes. Here. Yeah, I have one question.
Yeah.
Have you explored the production of ochratoxin A on the Aspergillus isolates? Because now in Europe, Aspergillus genus is considered the main responsible of the presence of ochratoxin A on wines.
No, I didn't actually. I didn't study, because in Australia this research has been conducted. In actually Australia, ochratoxin production is not really a threat, because ochratoxin is mainly produced by Aspergillus carbonaceus. Aspergillus carbonaceus has been found in Australian vineyards very, very minor, like minor levels. It is not a major species. Aspergillus niger is the most dominant species in Australian vineyards. I think Aspergillus carbonaceus is mainly found in Mediterranean region. Yeah.
Thank you very much.
Thank you very much.